Gut microbes eat our medication. The first time Vayu Mani rectal manipulated microbes, he made a decent sourdough bread. At the time, young Mani rectal, and most people who head to the kitchen to whip up a salad dressing, pop popcorn, ferment vegetables, or caramelize onions, did not consider the crucial chemical reactions behind these concoctions. Even more crucial are the reactions that happen after the plates are clean. When a slice of sourdough travels through the digestive system, the trillions of microbes that live in our gut help the body break down that bread to absorb the nutrients. Since the human body cannot digest certain substances, all-important fiber, for example, microbes step up to perform chemistry no human can. But this kind of microbial metabolism can also be detrimental, said Mani Rectal, a graduate student in the lab of Professor Emily Balskis and first author on their new study published in Science. According to Mani Rectal, gut microbes can chew up medications, too, often with hazardous side effects. Maybe the drug is not going to reach its target in the body, maybe it's going to be toxic all of a sudden, maybe it's going to be less helpful, Mani Rectal said. In their study, Balskis, Mani Rectal, and their collaborators at the University of California, San Francisco, describe one of the first concrete examples of how the microbiome can interfere with a drug's intended path through the body. Focusing on levodopa, L-dopa, the primary treatment for Parkinson's disease, they identified which bacteria are responsible for degrading the drug and how to stop this microbial interference. Parkinson's disease attacks nerve cells in the brain that produce dopamine, without which the body can suffer tremors, muscle rigidity, and problems with balance and coordination. L-dopa delivers dopamine to the brain to relieve symptoms. But only about 1 to 5% of the drug actually reaches the brain. This number, and the drug's efficacy, varies widely from patient to patient. Since the introduction of L-dopa in the late 1960s, researchers have known that the body's enzymes, tools that perform necessary chemistry, can break down L-dopa in the gut, preventing the drug from reaching the brain. So, the pharmaceutical industry introduced a new drug, carbidopa, to block unwanted L-dopa metabolism. Taken together, the treatments seem to work. Even so, Mani Rectal said, there's a lot of metabolism that's unexplained, and it's very variable between people. That variance is a problem. Not only is the drug less effective for some patients, but when L-dopa is transformed into dopamine outside the brain, the compound can cause side effects, including severe gastrointestinal distress and cardiac arrhythmias. If less of the drug reaches the brain, patients are often given more to manage their symptoms, potentially exacerbating these side effects. Mani Rectal suspected microbes might be behind the L-dopa disappearance. Since previous research showed that antibiotics improve a patient's response to L-dopa, scientists speculated that bacteria might be to blame. Still, no one identified which bacterial species might be culpable or how and why they eat the drug. So, the Balskis team launched an investigation. The unusual chemistry, L-dopa to dopamine, was their first clue. Few bacterial enzymes can perform this conversion. But, a good number bind to tyrosine, an amino acid similar to L-dopa. And one, from a food microbe often found in milk and pickles, Lactobacillus brevis, can accept both tyrosine and L-dopa. Using the Human Microbiome Project as a reference, Mani Rectal and his team hunted through bacterial DNA to identify which gut microbes had genes to encode a similar enzyme. Several fit their criteria, but only one strain, Enterococcus faecalis, E. faecalis, ate all the L-dopa every time. With this discovery, the team provided the first strong evidence connecting E. faecalis and the bacteria's enzyme, PLP-dependent tyrosine decarboxylase or TRDC, to L-dopa metabolism. And yet, a human enzyme can does convert L-dopa to dopamine in the gut, the same reaction carbidopa is designed to stop. Then why, the team wondered, does the E. faecalis enzyme escape carbidopa's reach? Even though the human and bacterial enzymes perform the exact same chemical reaction, the bacterial one looks just a little different. Mani Rectal speculated that carbidopa may not be able to penetrate the microbial cells, or the slight structural variance could prevent the drug from interacting with the bacterial enzyme. If true, other host-targeted treatments may be just as ineffective as carbidopa against similar microbial machinations. But the cause may not matter. Balskis and her team already discovered a molecule capable of inhibiting the bacterial enzyme. The molecule turns off this unwanted bacterial metabolism without killing the bacteria, it's just targeting a non-essential enzyme, Maini Rechtel said. This and similar compounds could provide a starting place for the development of new drugs to improve L-DOPA therapy for Parkinson's patients. The team might have stopped there. But instead, they pushed further to unravel a second step in the microbial metabolism of L-DOPA. After E. faecalis converts the drug into dopamine, a second organism converts dopamine into another compound, metatyramine. To find this second organism, Maini Rechtel left behind his mother doe's microbial masses to experiment with a fecal sample. He subjected its diverse microbial community to a Darwinian game, feeding dopamine to hordes of microbes to see which prospered. Egerthella lenta 1. These bacteria consume dopamine, producing metatyramine as a byproduct. This kind of reaction is challenging, even for chemists. There's no way to do it on the benchtop, Maini Rechtel said, and previously no enzymes were known that did this exact reaction. The metatyramine byproduct may contribute to some of the noxious L-DOPA side effects. More research needs to be done. But, apart from the implications for Parkinson's patients, E. lenta's novel chemistry raises more questions. Why would bacteria adapt to use dopamine, which is typically associated with the brain? What else can gut microbes do? And does this chemistry impact our health? All of this suggests that gut microbes may contribute to the dramatic variability that is observed in side effects and efficacy between different patients taking L-DOPA, Balskis said. But this microbial interference may not be limited to L-DOPA and Parkinson's disease. Their study could shepherd additional work to discover exactly who is in our gut, what they can do, and how they can impact our health, for better or worse. The evolution of puppy dog eyes. Dogs have evolved new muscles around the eyes to better communicate with humans. New research comparing the anatomy and behavior of dogs and wolves suggests dogs' facial anatomy has changed over thousands of years specifically to allow them to better communicate with humans. In the first detailed analysis comparing the anatomy and behavior of dogs and wolves, researchers found that the facial musculature of both species was similar, except above the eyes. Dogs have a small muscle, which allows them to intensely raise their inner eyebrow, which wolves do not. The authors suggest that the inner eyebrow-raising movement triggers a nurturing response in humans because it makes the dog's eyes appear larger, more infant-like, and also resembles a movement humans produce when they are sad. The research team, led by comparative psychologist Dr. Julianne Kaminsky, at the University of Portsmouth, included a team of behavioral and anatomical experts in the UK and USA. It is published in the journal Proceedings of the National Academy of Sciences (PNAS). Dr. Kaminsky said, "The evidence is compelling that dogs developed a muscle to raise the inner eyebrow after they were domesticated from wolves. We also studied dogs and wolves' behavior, and when exposed to a human for 2 minutes, dogs raised their inner eyebrows more and at higher intensities than wolves." The findings suggest that expressive eyebrows in dogs may be a result of humans' unconscious preferences that influenced selection during domestication. When dogs make the movement, it seems to elicit a strong desire in humans to look after them. This would give dogs, that move their eyebrows more, a selection advantage over others and reinforce the puppy dog eyes trait for future generations. Dr. Kaminsky's previous research showed dogs moved their eyebrows significantly more when humans were looking at them compared to when they were not looking at them. She said, "The AU101 movement is significant in the human-dog bond because it might elicit a caring response from humans but also might create the illusion of human-like communication." Lead anatomist Professor Ann Burrows, at Duquesne University, Pittsburgh, USA, co-author of the paper, said, "To determine whether this eyebrow movement is a result of evolution, we compared the facial anatomy and behavior of these two species and found the muscle that allows for the eyebrow raise in dogs was, in wolves, a scant, irregular cluster of fibers. The raised inner eyebrow movement in dogs is driven by a muscle which doesn't consistently exist in their closest living relative, the wolf. This is a striking difference for species separated only 33,000 years ago, and we think that the remarkably fast facial muscular changes can be directly linked to dogs' enhanced social interaction with humans." Dr. Kaminsky and co-author, evolutionary psychologist Professor Bridget Waller, also at the University of Portsmouth, previously mapped the facial muscular structure of dogs, naming the movement responsible for a raised inner eyebrow the action unit, O101. Professor Waller said, this movement makes a dog's eyes appear larger, giving them a childlike appearance. It could also mimic the facial movement humans make when they're sad. Our findings show how important faces can be in capturing our attention, and how powerful facial expression can be in social interaction. Co-author and anatomist Adam Hartstone Rose, at North Carolina State University, USA, said, these muscles are so thin that you can literally see through them, and yet the movement that they allow seems to have such a powerful effect that it appears to have been under substantial evolutionary pressure. It is really remarkable that these simple differences in facial expression may have helped define the relationship between early dogs and humans. Co-author Rui Diogo, an anatomist at Howard University, Washington, D.C., USA, said, I must admit that I was surprised to see the results myself because the gross anatomy of muscles is normally very slow to change in evolution, and this happened very fast indeed, in just some dozens of thousands of years. Soft tissue, including muscle, doesn't tend to survive in the fossil record, making the study of this type of evolution harder. The only dog species in the study that did not have the muscle was the Siberian Husky, which is among more ancient dog breeds. An alternative reason for the human-dog bond could be that humans have a preference for other individuals which have whites in the eye, and that intense O101 movements exposes the white part of the dog's eyes. It is not known why or precisely when humans first brought wolves in from the cold and the evolution from wolf to dog began, but this research helps us understand some of the likely mechanisms underlying dog domestication.